Welcome to our lab. Our lab focuses on basically plant molecular biology with a theme of agricultural biotechnology. We have many different projects funded both by external funds and not, that deal with using the modern molecular biology tools to improve modern day crops. The focal point in our lab has been for the past 5 or 6 years cassava, commonly known as yucca in Puerto Rico. But beyond yucca, we have different projects in lima beans, sweet potato, as well as the using of the model plant Arabidopsis. For research wise, one of the main projects that we have that is to assess transgenic plants in the field. Another project assesses the gene expression profiling of field-grown plants for different characteristics, genes involved in different characteristics. Another project has usage of the SSR marker technology to assess the genetic diversity of both cassava in Puerto Rico as well as sweet potato in Puerto Rico. One of the most novel projects that we have right now also tries to see the expression pattern as well as the functionality of two novel genes isolated from cassava. These two genes are actually in the detoxification pathway of cyanide, where a previous graduate student a few years ago who worked in our lab isolated the two genes, and now a new graduate student is trying to express them in model plant Arabidopsis to see its functionality, whether it is involved in which part of the pathway of cyanide detoxification.
My name is Lorraine Rodriguez. I'm a first-year grad student here in the lab, and my work is related to assessing the genetic diversity of sweet potato using SSR markers and also using a fluorescent technique called M13 primer that allows us to identify different clones and different accessions from different plant varieties from the ones that we have either on tissue culture or around cultivars in the island.
Hi, my name is Kevin Babylonia. I'm working with the characterization of two genes from cassava. Those two genes are the beta-cyanoalanine synthase gene and the cysteine synthase gene. Recently, those two genes were isolated in this lab, and apparently the beta-cyanoalanine synthase is involved in the cyanide detoxification pathway, and the cysteine synthase is involved in the cysteine synthesis in cassava. So those genes were expressed in E. coli, but E. coli is a bacteria and those genes are from a plant. So right now I'm expressing the genes in Arabidopsis thaliana, that is a novel plant used for molecular genetic studies.
Hi, my name is Andres Salcedo. I'm a grad student of Professor Siritunga, and my work is focused in the gene expression analysis of physiological post-harvest deterioration in cassava. This endogenous disorder affects the alimentary security for millions of people around the world, and also affects the expansion of cassava as a crop. I'm so happy to share this interesting opportunity of research in the University of Puerto Rico.
So this is some yucca from a recent harvest of a project where we are trying to improve the nutrition of cassava for Africa. And in this project, the target is to genetically engineer the plant to have higher protein, higher vitamin A, beta carotene, and high iron. So in fact, this yucca that was harvested last week has high proteins, about 5 times more proteins than found in normal cassava. Furthermore, this yucca has also been engineered to have reduced levels of cyanogens. So it's two-pronged attack to reduce cyanide, but at the same time increase the protein content of cassava. The idea of this project is to plant these experimental plants in agricultural field stations in Puerto Rico under controlled environments and assess the agromorphological characteristics compared to wild type cassava. Once experiment is done, the top 1 or 2 lines per trait will be planted and experimented in soils in Nigeria, and after that it will eventually be released to the public free of charge.